Okay, good afternoon, everybody. We still have a few people filtering in here in the room, but we are going to go ahead and get started. So thank you, everybody, and welcome to the Great Lakes Seminar Series. We are pleased to welcome Dr. Charlyn Partridge today. She is an associate professor at Annis Water Resources Institute at Grand Valley State University, and her research uses genetic and genomic tools to aid in conservation and management efforts. Her current projects involve using eDNA approaches for targeted species detection and understanding how invasive species rapidly adapt to new environments. So welcome, Charlyn.
Okay, first of all, this classic Zoom question, but just for the people in the room, can everyone hear me? Awesome, thank you. So first, I just want to say thank you for the invitation. This talk might be a little bit different than what you're used to because I'm bringing everyone to the terrestrial system instead of the aquatic system. And I'm going to talk about one of probably the biggest threats that we currently have to Michigan's forests. So imagine you're on a weekend trip, you and your family, your friends, maybe just, if you're like me, just you by yourself because you like to hike in the woods alone. No danger in that. You're walking through the woods, you're on this gorgeous trail. You look to your right and you see something that looks like this, where you're on the trail, there's nice light shining down, but then you see this wall of dark. What do you think is behind that wall of darkness? If you were to walk through this little entrance right here, in part, where would you come out? Does anyone know? I can tell you when my students and I are out in the field, this is what we're looking for. When we are out in the field doing our work, we are looking for spots of darkness in a forest. If you were to walk through this little archway, you would come into a hemlock stand. So eastern hemlocks drive these unique ecosystems because of their characteristics. So eastern hemlocks have very dense canopies and very thick canopies that will block out light in that system that limit understory growth. So going just a few steps from this side to this side basically limits any understory vegetation. Also, because of that very thick canopy, when you walk into a hemlock stand, you can physically feel the change in temperature. It drops by like 2 to 3 degrees to where you might have to put on a light jacket. So not only does it change those light and temperature properties, but because of the physical nature of the hemlocks, because of its construction, it regulates soil chemistry. So things that live there live in the soil are more related to acidic environments. And it can also, because of its evapotranspiration rates, it can regulate the amount of water yield within a system. So hemlocks, specifically eastern hemlocks, are what we know as foundation species. So because of these characteristics, they regulate these environmental parameters pretty strongly, things like temperature, light, soil chemistry, and water yield. So they create these little microclimates that house a unique biological community within these ecosystems. And the reason why we care about them in Michigan is because we have over 170 million eastern hemlocks within our state. So especially as you kind of move up into the upper peninsula, you have one of the highest densities of eastern hemlock that we see anywhere within the United States. So these are very important communities that have really strong impacts on what happens to be present within that ecosystem. However, this ecosystem is currently under threat by a tiny invasive species known as hemlock woolly adelgid. So like I said, these guys are pretty tiny. So this individual here is what's known as a crawler. So this is a crawler stage. These guys are less than a millimeter in size, roughly the size of a poppy seed. And basically what will happen is they will go and they will land on new hemlocks. They will go through various stages and develop into an adult that looks like this. These individuals have a very long mouth part called a stylet that they will insert into the twig of a hemlock. They will feed that stylet into the nutrient-rich cells, and they will actually suck nutrients from a hemlock. And this results in hemlock mortality within 4 to 10 years. If you imagine what hemlock mortality will do to an ecosystem like a hemlock forest. So this is an image of a hemlock tree in one of our sites. So here's the trunk, and then here's the broken top on the ground over here. And this image is basically from me standing at the base of that hemlock tree and just taking a picture straight up. And kind of what you can see is this big open space that's now being created by the death of that hemlock. So now where that was normally blocked, blocking out light, any type of radiation that's coming down, or solar radiation to kind of increase temperature, that is now wide open, right? And so now that allows light in, that causes now more understory growth to happen. And in areas where we know that these ecosystems have been impacted the longest, like there's a long-running study in the Harvard Forest looking at HWA's impacts on hemlock stands, we see a shift in the vegetative communities in these systems. So you move from like these coniferous trees to things like bushes and shrubs and more deciduous trees. So not only are you changing the vegetative community, but because that vegetative community changes, the insect community changes, the bacterial community can change, the bird community can change. So you have these cascading level effects when you lose these eastern hemlocks. And in many areas, specifically in eastern North America, it's not the loss of one hemlock, but some of these stands have lost up to 95% of the hemlocks within these areas, and these are known as ghost stands. So this is an image of a hemlock forest where all of these brown trees are dead hemlocks. So you can imagine at a large scale how large-scale loss of hemlock is really going to impact and alter these systems at an ecosystem level change, right? Oh. So where did HWA come from? How did this invasion start? Well, what we know is HWA was first detected in around the 1950s near Richmond, Virginia. There's a little blue dot somewhere in here, right around there. And from that time, it has rapidly expanded throughout the majority of the eastern hemlock distribution range. So we have invasive populations all the way from Alabama up to Nova Scotia, and then as far as west as Michigan. And kind of like these green dots are the hemlock distribution in the US. So since roughly 70 years ago, these populations have rapidly expanded throughout the majority of the eastern hemlock distribution range. And the only thing that really is keeping it at bay at these northern extremes is cold temperature. So die off due to high winter mortality. So when we look at where these invasive populations came from, so right here is a phylogeny of HWA based upon a 669 base pair region of the cytochrome oxidase gene. And it's looking at native populations. So we have populations in China, Taiwan, Japan. This is a small island off the coast of Korea. We have our North American invasive populations, and then interestingly, HWA is also native to Western North America. So populations have been in Western North America from anywhere between 10 to 40,000 years. And when we look at our invasive Eastern North American population, what we see is we have a haplotype that's present that groups in with the Southern Japanese group. And so, or Southern Japan group. And so the hypothesis, or what we think based upon the molecular data, is that this invasion initially happened due to infested nursery stock that came from Southern Japan hemlock that was imported and then was planted in Virginia and then has since spread from that time. So one of the really, really interesting things about HWA, so I'll go back and forth between hemlock woolly adelgid and HWA because hemlock woolly adelgid is really hard to say 50,000 times within an hour. So is that their life cycle can be really plastic. So there are populations that have both asexual and sexual life stages, and there are populations that just have asexual life stages. And so what happens with those populations, mainly in the native region, that have asexual or that have both stages, that you'll have an egg that will hatch into a crawler. The crawlers are one of the most mobile phases of hemlock woolly adelgid, so that's where we get spread among hemlocks. And then they'll go through a couple of different instar nymph phases, lay eggs, go through another couple of instar nymph phases, and then they'll produce winged adults. That winged adult will then go and move from a hemlock to a spruce. So sexual reproduction only occurs on spruce trees. Go through a couple of metamorphoses. That individual will then or the egg that's laid by the winged individual, some will hatch into females, some will hatch into males. Some individuals may produce both males and females. We're not exactly sure how that happens. You will have mating between a male and a female. Those individuals will produce one egg. That one egg will then develop and produce, start to feed and produce a gall on that spruce. Then they will lay hundreds of eggs into that gall that will eventually break off, form wing insects that then move back to the hemlock. So for those populations that have this complete life stage, this takes about 2.5 years to happen. But in North America, we only have this asexual stage. So individuals will develop from eggs to crawlers, then go through these instar nymphs, do it again, and then recycle back. So we have roughly 2 generations per year, but all of the populations in North America, both Eastern North American populations and Western, the native Western North American populations, are all asexual. So, and that kind of has an impact in terms of rapid population spread, because we know and studies have shown you only need one new individual in a new area to create a new invasive population, right? So you don't need a set population density for that population to then take off. So what about HWA in Michigan? Well, our current infestation was first detected in 2015 in Allegan County. There were earlier detections, but these were thought to be eradicated. So there was one in Sleeping Bear Dunes in like 2006. It was a single branch of a tree and they removed it and burned it. But our current infestation is thought to have started from this from this Allegan County population. Since then, it has spread through most of the counties in the western part of the state. So this inset map shows all of the counties where it's currently been confirmed. This map here, even though it was produced in June 2025, it is already outdated. We have multiple known infestations in Manistee, and our current known infestation in Antrim is just south of the border, the county border. So one of the things that our lab does is we help with monitoring of hemlock woolly adelgid in high-risk sites. So these are sites that have been defined by our partners as sites that they think HWA could potentially be at, and they want to monitor more extensively, but HWA has never been found there yet. Okay? So how do you typically monitor for hemlock woolly adelgid? Well, the way it's typically done is you send out you send out a crew into the field. And what individuals are looking for, they're looking for these little white, what we call ovisacs. So this is material secreted by the adelgid that they then lay their eggs in. And then the individuals in the eggs can overwinter and then protect them from the cold. But they're basically material secreted by the actual adelgid itself. And so you will have crews that will go out in the fall and winter and look for these little tiny white sacs on the base of a hemlock needle. So can anyone tell me a problem with looking for little tiny white sacs on the base of a hemlock needle in the middle of winter in Michigan. Snow, right? So it's not always easy to see because trees are covered in snow. So that's one issue. Number two, we have 170 million eastern hemlocks in the state. If you have to look at every single tree, that will take a very, very long time. And a lot of times these infestations start at the top of the canopy and work their way down. Most of the time when individuals are surveying, they're grabbing branches at arm's reach, right? So if you're starting an infestation at the top of the canopy, you may not see that until the infestation has already kind of been there for a few years. So what our lab is doing is we are using airborne eDNA to kind of help with these monitoring efforts. So I know many of you work with aquatic DNA. Same thing, same principle, just like you get DNA from fish sloughing off cells and mucus or excreting. So I like to think I do a lot or do some outreach with K through 12 groups at AWRI. And when I talk about eDNA and airborne eDNA, I tell them to imagine themselves as Pigpen from the peanut strips that just has that cloud of dust, but you're surrounded by a cloud of your own DNA. Right? Every time you scratch your arm, you're releasing DNA. You cough, you're releasing DNA. So we're all just like bags of flesh walking around with a trail of DNA behind us. And so we can capture that DNA from the air and then test it to see what is there. So these are the traps that we use. We design them specifically to be easy to use and very low cost to make. So they're 3D printed traps. They have a base and a lid. And then they hold 4 microscope slides, and those microscope slides are coated in petroleum jelly. So literally just dipped in melted Vaseline. And then they're dried, and then we set them out in the field, and then anything that lands on those slides get captured. We bring it back to the lab, extract the DNA, and test it for hemlock woolly adelgid DNA. And this is just what one of those traps looks like when it's deployed. So we've been doing this since 2022. Like I said, these are placed in areas that are high-risk sites with no current known infestation. We put them out from May to June. This is during the active crawler period. So remember I said the crawler is the mobile phase. So a lot of times we can pick up crawlers, but a lot of times we also just pick up HWA-associated material, ovisac material, a leaf that an HWA individual or a needle that an HWA individual may have sat on. But we deploy them from May to June because that is the most active crawler period, and that increases our chance of capturing something. We exchange those slides every 2 weeks, and then we're targeting a 250 base pair region of the cytochrome oxidase gene and using qPCR to test for presence absence. And this is kind of like what a slide will look like when we bring it back from the field. So one thing I will say is because these individuals are so small and the amount of DNA that they produce is so small, these have been very useful in the fact that it limits bycatch of anything else, right? So you could put kind of a bigger trap out there or sticky trap, but then the bycatch becomes so much that you miss potentially the HWA that's present on there. So this was specifically designed to kind of capture small things. So this is just kind of an example of our process. So we have our slides. We basically scrape the petroleum jelly off those slides, put them into a bead bashing tube. We do repeated heat cooling centrifugation cycles to kind of separate what was on the trap from the petroleum jelly. So the material from the petroleum jelly, we extract it, and then we run all of our samples in triplicate and then analyze it for presence absence. So the way we determine a positive detection is a little bit different than how many eDNA groups determine positive detection or no detection. For us, all 3 of our replicates per sample that we run have to test positive for HWA before we tell our partners that there is a positive detection. And that's because when we tell our partners there's a positive detection, that actually results in direct action. If we say we got a positive hit here, that goes on their list to survey in the fall. And so we don't want to accidentally tell them it's there when there's not, because that's time, money, and effort that our partners have to spend if we're not confident in that detection. So when we say detect, we are 99% positive that there is a detection at that site before we ask our partners to send crews out to look for it. And this is just kind of what those detection results look like. So we have a target threshold, and then this would be a positive trap. So our 3 replicates come back positive. This is a positive trap. And then all of our samples are run with an HWA positive control. So it's a synthetic G block that we use. So how has this resulted so far? So from 2022 to we've been doing this again from 2022 to 2025. And the first year we set out 55 traps. 2 of those traps came back positive and 1 new stand. So it was 2 traps in a single stand that came back positive. And 2023, 4 of our 55 traps came back positive, and this was 3 new stands that came back positive. We didn't have a grant for 2024, but we were contracted by a group to run 25 traps for them. One of those came out to be 2025, or one of those came out to be positive. And then for 2025, we greatly expanded where we were surveying. So this past year, we have traps all the way from Manistee County out into central portions of the UP. So we deployed 235 traps, and 12 of those came back positive. We had traps come back positive in Manistee, Leelanau, Antrim County, and Benzie counties. That all came back positive. And now kind of the current most northern known stand that's infested in Antrim County was initially detected by our trap, which was then gone out and surveyed to find, and they found the infested tree. So we think this works pretty well. Out of all of our traps that came back positive, they have gone to survey and they have found the infestation. So we're pretty much, I guess, shooting like 100% in this. And these 4 years of data tell us, and the fact that it's constantly found every single time we say we got a positive detect here, is that, you know, we're pretty confident in what these results, and pretty confident telling our partners that when we get a positive detect, it's likely going to be there. And this is just a map that came from our partners at Huron-Manistee, showing kind of like, this is from 2022 and 2023. So in 2022, the yellow triangles represent where our traps were that had positive hits. And then when they surveyed, the pink stars represent the trees that they found that were infested. For 2023, the red triangles represent our traps that came back positive. And then the green circles represent where the trees were that were infested. So we have pretty good detection success for this project, or for this method. One of the problems from talking to our partners is that going out and exchanging slides every 2 weeks can be problematic, especially like one of our partners is Sleeping Bear Dunes, and they survey on the Manitou Islands. Going out to North and South Manitou Island, virtually not a thing. They can do it for a couple weeks, but normally we ask for a 6 to 7 week collection period. And so we're also looking at the use of, or potentially using spider webs as passive eDNA devices for capturing forest pest. We did a trial study last year. They work really, really good in infested areas. Every single spider web we collected in all of our known infested areas came back positive. How well they work in high-risk sites has been a little bit variable. All of them came back positive in Huron-Manistee. All of those were known, or all of those ended up being infested. We missed a couple in Antrim and Leelanau. And so we're trying to figure out why our traps found it, but the spider webs didn't necessarily find it in some of those infested sites with very, very low levels of infestation. Okay, so that's how we use it to monitor in high-risk areas. And many times, like I said, for these methods, like that is presence absence. So we're not really trying to quantify how much DNA is like in the system. And but there can be some uses, right, for and most of the time, like our partners don't care what quantity of DNA is there. They want to know if it's there, because if it's there, they need to treat trees. If it's highly infested, low infested, they still need to treat trees, right? But there are some cases where knowing the infestation level of a stand may be important. So you can imagine a case where now where the infestation has spread so far, you cannot treat every single stand. So then it might be beneficial to know, is the stand highly infested? Is the stand have a low concentration of infestation? Do I prioritize treating highly infested sites to try to knock that back? Or do I prioritize sites that I think I'm going to eradicate it from that site? So those are the kinds of decisions with that information that managers can make. Also, the treatments that are currently used, the chemical treatments, aren't lifelong treatments, right? So they're two different treatments. One lasts from 2 to 3 years, one lasts from 5 to 6 years. So you can imagine areas where they've treated, those may need to be retreated 5 years later or 2 years later. Do you just treat it 5 years later, or is there a way that we can quantify that infestation, see when that infestation is going back up again or tending to go back up again, and then treat it instead of just arbitrarily picking a date? So in those cases, we see that quantifying may be useful to assess these infestation levels. So to see kind of how well these methodologies help capture how infested a site is, we looked at HWA infestations to see if we could correlate qPCR values with how many HWA individuals are on a tree, right? And this was work done by my former grad student, Keely Dunham. So we took 9 sites of known infestation. These were in Oceana and Mason County. The sites ranged in different infestation levels. So some had very low levels of infestation. Others had very high levels of infestation, and they had different years of treatment that kind of correlated with some of that infestation level. And then what we did is, so if you want to measure an infestation of HWA in a forest, what you have to do is you have to go to that stand, you have to pick a random number of trees, then you have to pick a random number of branches on that tree, then you have to count a number of individuals that are on like the end of that branch for a set distance. So like we picked the number of cystins on 100 new growth needles, right? We had 9 sites. 9 sites, 10 trees per site, 9 branches per tree. This takes a really long time. This is at the minimum end of the numbers of trees and branches you would have to sample. Most groups do 50 trees per site. I had a 2 years master's student, so timing is a thing, right? And 9 sites. So this takes a very long time. This is not something that you can do in an hour, right? This is days and weeks of work. So we want to see how that random branch sampling method compared, so the traditional way of measuring infestation compared to us using our traps to capture adelgid crawlers and just count the number of crawlers that are there. And then also using qPCR analysis on those slides to kind of see how that correlated. So our idea was, can we use these crawler counts and these qPCR values to give us an estimate of how many cystins would actually be on those trees? So just kind of to orient you to HWA life history. So what we're measuring, the cystins are the individuals that are actually settled and feeding on the tree, or they are the individuals that these individuals, if I can get this in. So these individuals. So the crawlers have gone through a couple of instar phases, and now they're kind of like settling and getting ready to overwinter. So those are the individuals that we're counting on the tree. And then the crawler counts are basically these guys. So they are the offsprings of the cystins. So these guys are the babies of these guys. Does that make sense? Okay. So For the cystin counts, we went through our 9 sites. In fall and winter 2022, we counted 10 trees, 9 branches on each tree, and the number of cystins that were present on 100 new growth needles on each of those branches. For crawler counts, we deployed 5 traps per site. They were deployed from April to August, and exchanged every 2 weeks. That was for a total of 7 collecting periods. So 7 2-week collecting periods. And then we would take these individuals, scrape off that petroleum jelly, extract the DNA, and run that with qPCR analysis to quantify how much DNA there was. So we would expect, since these are the offspring of these, this should correlate highly with that. And since this is literally the DNA that's coming from this, that should correlate highly with that. Makes sense? Okay. So what did we find? So when we look at our crawler counts relative to our qPCR values across all 7 of our sampling periods, across all 9 of our sites, this is a relationship we're looking at. So on the x-axis, we have our crawler counts, and then on the y-axis, we have our gene copies per reaction. So if you're getting exactly the amount of DNA out of every single individual, and there's a direct correlation, we would expect a direct line of 1, right? And so we can see that positive relationship. I mean, we have an R-square of 0.73. It's significantly different than 0, but there is definitely some noise happening around it. But there is a good, strong correlation. More often, when you have lots of individuals on your slide, you're going to get lots of DNA back. If you have a few individuals on a slide, you're going to get a few individuals, or a little bit of DNA back. However, when we look at this relationship and we break it up across our 7 sites, what starts to happen is that relationship changes a little bit. So some areas we still see, a pretty strong correlation, but then other areas that correlation starts to get smaller. So 0.23, 0.33. And what we're typically seeing across our sites are those low infested sites typically have lower correlations than our high infested sites. So we think that's a couple of reasons. We think that DNA extraction efficiency is a problem, and that becomes more noisy when you have a low number of individuals than when you have a high number of individuals. We also think because not only are we collecting crawlers, we're collecting eDNA. And if you have eDNA at your low infested sites, that is going to make a lot more noise within those samples compared to what you would see at your higher infested sites. So we think both environmental and technical aspects of just our methods are kind of causing more noise, and that noise is being amplified higher at those low infested sites compared to some of those high infested sites. So across the board, there's that positive relationship, but it starts to break down a little bit at those low infested sites. So this is, again, just between crawler counts and qPCR values, which should be extremely tight, right? Because it's literally the DNA from the slide that the crawlers are on. But our question was, can we actually correlate these crawler counts and these qPCR values to cystin counts, the number of individuals that are on the tree? That is the classic way of estimating estimating HWA infestation levels. So for this, what we did is we solely focused on the peak crawler period. So if you imagine the crawler period is when the cystins eggs that they laid are hatching into crawlers. And you can imagine this starts in late June and kind of ends in mid-June. But you get this peak period where the majority of those eggs are hatching and the crawlers are out, right? So that's where, if we plotted number of crawlers that we see over time, it looks like boop, boop, right? So what we're doing for this day is we're just looking at that peak crawler period, because that should be our highest correlation to the number of cystins. So, so this data is just from that peak crawler period. Typically early June, the window does shift a little bit from year to year, but late May to early June is typically where it is. And so this is looking at crawler counts versus cystin counts, so the number of offspring on the slides compared to the individuals that birth those offspring. And we get a really strong correlation. And then this is cystin counts compared to qPCR values. Again, a pretty strong relationship when you focus just in that 2-week period of the peak crawler period, as opposed to the whole 7-week period. So you don't have to collect all 7 weeks. You can just focus on that 2-week peak crawler period, get your data from there, and that will correlate very well to your cystins counts. So when we did this, again, we worked very closely with SISMAs, National Park Service, tribal groups, with the data that we share with these individuals. And so a lot of the questions are like, those groups don't have thermocyclers sitting in their lab, right? They may not have the money to pay for thermocyclers sitting in somebody else's lab. So the cool thing that we found about this is that the relationship for compared to cystins counts was just as strong for crawler counts as it is for those qPCR values. So what that means is you can put a $5 trap out in a field, leave it there for 2 weeks, go back, collect those slides, count the number of cystins on that slide, and in 1 day, you can have a good estimate of probably how infested your site is relative to the weeks upon weeks of time of going out, measuring 50 trees, counting tens of thousands. I actually calculated how many cystins my student counted, and I can't remember, but it was like in the tens of thousands of cystins. So for that actual random branch sampling. So this is something that we think is helpful for our partners because it's cheap, it's fast, and it's easy. And is it going to give you exactly the number of cystins in a system? No, but only sampling 50 trees of 1,000 is not going to give you the exact number of cystins in a system either. Can it give you a proxy to tell you there are few or a lot you probably need to worry about this site, or this is a newly infested site, or kind of give a little bit information that can help them to prioritize their management plan? Yes. So that's kind of what we kind of took from this, is that it can be used to help those management decisions, but then also you can track to see if you see a trending increase in crawler numbers over time, and that can trigger reapplication for treatment for any of these sites. So that's the work that my lab has been doing starting since like 2018. So what I'm going to talk about now is some of the future work that my lab is moving into. And so I will tell you, when I say future, I mean literally we started in January and it is now February 18th, right? So just kind of prefacing that. But what we're starting to look at is whether or not we can use population genomics to kind of help us understand this invasion a little bit better. And this is work currently being done by my master's student, Grace Forthouse. So questions that we want to ask is, can we use genomics to explore population structure of HWA throughout its invasive range? Because remember, these are all clonal individuals. Basically, they reproduce asexually through parthenogenesis. So can we use a genomics approach to kind of look at genetic variants and how they may be spreading? And then can we identify patterns of population divergence as HWA is rapidly expanding? So and this is just a map of a reminder that these infestations started in Virginia and kind of have been spreading out since that time. So you can imagine this continuous expansion, but then we also have some of these disjunct populations like Michigan. We have a big jump to Michigan. They literally crossed water to get to Nova Scotia. So what is happening there? Like, are those still continuous invasions, or did we have a new invasion from infested nursery stock? And so what we're starting to explore are trying to understand these patterns as they kind of move out through the system. Again, trying to understand if it's just been like this continuous expansion of as gradually as they moved out, and there's really no difference between an invasive population in Michigan compared to an invasive population in Georgia. Maybe there's just such rapid expansion, there hasn't been enough time for population differentiation. Or, so we talked about cold temperature being a primary pressure for mortality. Maybe strong selection pressure in more colder climates may be selecting for cold-resistant individuals, and there is some suggestive evidence that individuals in northern regions have higher cold tolerance and can keep that cold tolerance longer than individuals in the southern region. So is there adaptation happening in some areas compared to others? Are we just seeing drift over time as you get these small founder events, and then just drift is causing population divergence across these regions? So those are the kinds of questions we're trying to answer. So we sampled 150, so last summer in spring, well, when I say we, I mean Grace sampled in Michigan, and then our amazing partners throughout the US Forestry Service sampled everywhere else, just like they're currently doing for us right now. But we sampled 115 individuals throughout Georgia, North Carolina, Virginia, New York, Nova Scotia, and then Michigan. And then we also were able to get samples from their native range, where we think the invasion initially came from, in southern Japan. And then we did low coverage whole genome sequencing, and based on the data that we got back, that results in roughly a 5x coverage of the genome. So the genome's about 220 megabases. And this is just a map with the black dot showing kind of the distribution of where we've sampled from. So again, literally we just got this data back. So this is the most preliminary data you have ever seen in your life. Like you're the first people other than like Grace and I to have looked at it. So we're still trying to play with it to figure out what it means. But when we look at our populations with Japan included, so basically what we did is we identified roughly 46,000 SNPs. So these are single nucleotide polymorphisms. So they're genetic differences throughout the genome between individuals. And what we're seeing is that we have two main groups from where we sampled in Japan. So 4 individuals clustering together, then another 4 individuals clustering together. Some of that is based upon where they're sampled from, some of it is not. But then when we look at North America, they're all grouping together in a tight cluster. Which is what we expected, right? So you're like, this was our control, like, did we do it right? Like, theoretically, the ones in North America should look different than the ones in Japan, because it's been 70 years since they've been with one another. So this was like, yay, this explains what we were looking at. But then kind of what we wanted to see is, again, our hypothesis was that over 70 years, as these populations have expanded, we should get little founding events, and then we should see population differentiation due to drift. So populations, if I would predict, populations in Nova Scotia would look and cluster separately from populations in Georgia, right? There's enough distance, it's been enough 70 years as they've moved out there. That's what we would expect. And what we find is this, is that basically there's no real population structure based upon the 44,192 SNPs that we've seen that we identified within our North American cluster. The good news is that this does suggest that the initial hypothesis that this was probably a single invasion event matches. So our data match with that. This was probably a single invasion event. But what we're trying to understand now is kind of like, why is everyone clustering together despite those very drastic, like mild differences, right? So the physical difference, why are these still all clustering together, and what is driving that grouping? So one of the things from talking to our collaborators, this is both, so HWA does have sex chromosomes, so this is with autosomes and sex chromosomes included. So he was like, aphids do weird things, and they have really cool things with sex chromosomes. Maybe just look at the sex chromosomes. So I was like, awesome, we will just look at the sex chromosomes because that might be interesting. And then when we look at the sex chromosomes, we see the exact same pattern. So this is literally our baby first step of exploring this data. We're looking at genetic diversity across these populations. I can tell you that most of the genetic variation that we're seeing is likely going to be due to heterozygosity. So these guys are asexual. So most of the mutations that we're seeing are going to be novel mutations, and they're present in heterozygosity format. So we're probably going to see excess in heterozygosity of these populations based upon like locus level heterozygosity values. But we're kind of looking at, okay, we know they're all clustering together, but do some populations have more genetic diversity than others? What might be driving that? We also have treatment information for all of these sites as well that we're also going to apply on top of it to see if that does anything with the data. So where are we going? Like I said, baby, like literally hot off the computer or hot off the cluster for those data. But where are we going next? So what are we going to be looking at in probably over the next 3 years in my lab? So we not only are we going to be looking at genomic structure within the invasive populations, but we're also going to be comparing genomic structure of the invasive populations in Eastern North America to the native populations in Western North America. Those populations have very different selection pressures. In Eastern North America, the only selection pressure is cold temperature at the top part of that invasive range. The Western populations have native predators that basically back down or knock down those populations consistently. So there's very strong selection pressure that populations in Western North America see that Eastern North American populations do not. So we're interested in understanding how differences in that selection pressure is driving genetic structure and how just this rapid population expansion and rapid population growth is changing, altering the genomic structure in the invasive populations. Like I said earlier, there is some suggestive evidence that populations in the north, despite lacking of any clustering, are adapting to colder temperatures. So next winter, I'll be working with colleagues doing transcriptome analysis, where we take individuals from the north and south of the invasive range, expose them to cold temperatures, and look at transcriptome differences between those. And then that is also being done at the same time with individuals at UBC looking at Western populations. One of the other cool things that we don't really have time to talk about is HWA also has these microbial endosymbionts that help with kind of like the nutrient breakdown from the hemlock. And because these guys are so tiny, when we sequence or extract the DNA from the individuals, we're extracting DNA from whole individuals. Like we can't break off a leg and extract DNA because you can't see the leg, because it would be microscopic. So we not only are we getting sequences of the HWA, but we're getting sequences of all those endosymbionts as well. So we're going to have HWA individuals and their endosymbionts and kind of what trees they're found on. So eastern hemlocks and western hemlocks, so the individual tree species. And we're going to be looking at relationships between how those endosymbionts may change relative to what they're actually feeding on. So that's just kind of like a small glimpse into the future of the work that we are actually doing. And with that, I would like to thank all of my lab, because I am the one sitting on the computer and they are the one in the field doing all of it. And then I walk in the lab and I go, is everything fine? And is anything on fire? And they say, yes, it's fine. Nothing's on fire. And I go, good job. And then I go back to my office. So it's their workforce that's doing it all. And then also so many of our partners, the National Park Service, all the local SISMAs, I couldn't actually fit all the local SISMAs on here. But the tribal groups that we work with, everyone that we collaborate with over the summer, are collaborators with the Forestry Service in Canada and the Canadian Forest Service, DNR, and then funding from Michigan Space Grant, NSF, and the Michigan Invasive Species Grant Program, and also Forest Service as well. So with that, I will take any questions.
Thank you so much, Charlyn. I would be happy to bring the mic around to anybody who has a question.
Hey, thank you. That was great. So it looks like HWA has not hit the Upper Peninsula of Michigan yet. Is that inevitable? Because I also saw the Northeast had some spots that it didn't hit and there's no barrier. like the Great Lakes.
Right. So I will say that we are monitoring it very, very hard because we think there is a good chance that it will eventually reach the UP. So the question, like this winter, I think is great. Like we need to have more winters like this, but harder and colder because that knocks it back. So probably what we'll see next year is maybe not as much expansion based upon kind of like the data that we've looked at in the past. But if we keep warmer winters, then, I mean, the crawlers spread on the wind. Like they spread on birds, they spread on, so yeah, unless you put a big like barrier so that birds don't fly over it to the UP, which I'm like, let's do that. So yeah, if we have a lot of warmer winters, then yeah. But I will say there are many, many teams surveying and we have traps up there. So, but yeah, that's our fear. Is that it reaches the UP. Just because the quality of hemlock up there is so amazing and the density of hemlock is so amazing. So, yeah.
Thank you.
I don't want to be a naysayer and be like, it's inevitable, but
I was wondering what the HWA eradication process looked like and what kind of like the success rate was for eradicating in certain areas.
So if you have a very small population, like I said, it was found in sleeping bear in 2006 and they eradicated it, but it was literally one branch, right? And they just like, and moved it off. So there are two different current chemical treatment options. One is a fast acting but short-lived option. So it'll kill the HWA within a year, but only lasts like 3 years, I think. It's kind of, Demacola may have data saying that it's a little bit longer than that, but it's like that. Imidacloprid is the other option. So it does not act as fast. It takes about a year for us to see effects, but then it can last for 6 years. So if you get a site that is freshly like invaded, it works really, really well. The problem is stopping new infestations from moving in. There's also been some experimental work in the Eastern North America, specifically around New York, looking at natural predators from the West because we have HWA in the West. So they've taken some of the predators from the West and moved them over. There's been so I think the data for that's a little bit mixed. Like they will feed on HWA. The question is whether or not they can feed enough to knock back the HWA populations and how long it takes for them to actually get settled in that space once you actually move them over. So those are kind of the treatment options right now. Chemical and natural predation are kind of the things that are being explored. Like I said, chemical treatment has been great, but it takes time, effort, and money. So honestly, if you ever meet anyone who says that they have been part of an HWA treatment crew, buy them a beer because those crews, they're literally carrying backpacks of imidacloprid. They are like climbing ravines with hemlocks. They are drilling holes in each single hemlock, and then they are ejecting imidacloprid into each single tree. So anytime a student is like, oh, I've worked on an HWA treatment crew and I'm interested in your lab, I'm like, yes, please come. Because the amount of work and effort it takes is a lot, but there are limits in how much you can apply within a certain area. So a lot of times it's not the amount of like person power who are willing to go out there and do it. It's that a site will reach that limit and they just can't treat any more trees because they've reached that limit. And sometimes that limit gets waived to kind of help them treat more, but yeah, it's a lot of work to do it.
Thank you.
Sure.
Okay. Margaret, do we have any questions online?
We have a couple questions. First one, great talk. Can you speak more about the nutritional microbial symbiosis of HWA? Any idea how those relationships may or may not change seasonally or geographically?
Yeah, that's a really good question. It's something that we just started just because of the fact that we have the sequence data. So we thought, oh, this would be, because we had all of this data and realized that only a proportion of it was aligning to HWA. So we had all of this data and we were like, well, what is that? So I blasted it and then it was like, oh, the endosymbiont of HWA. And I'm like, that makes sense. Thank you. So they have two different endosymbionts. They actually have special like organs within their body that houses one endosymbiont and the other is kind of like free living. We do know that different species of adelgids have different species of endosymbionts. I don't think it's really clear as to how it will change by geographic region. My hypothesis is that we should see some variation because in different geographic regions, different HWA feed on different hemlock species that are chemically different. So I think that we would see some variation where we would see kind of clumping of endosymbionts from species who feed on western hemlocks versus eastern hemlocks. I don't know how it would change over time. We haven't really thought, or within a year, we haven't really thought about that. And I haven't really looked into that. But I do expect us to see some regional differences, but just because of the fact that there are species-specific differences and the different species feed on different trees as well. So what defines an HWA species is what its host tree species is.
Thank you. The next question is, what is the predator that controls native populations?
So there are a couple, so that's a long answer because there are a lot actually. And I am going to get their names wrong, so I apologize. One of them, they're nicknamed Luca and Lenny. One is a beetle and one is a fly. And so, but those are the two that I think that they've seen most promise with, being able to kind of rear them and easily release them. But I know in the Western United States and then also in Asia, there are a large number of predators, but they're specifically for management efforts, they're two that the research groups are focusing on. Oh.
I have another one.
If there's any. Okay, great. Well, then we will thank Charlyn for a wonderful talk.
Did you have one more?
I'm so sorry.
Yeah.
I'm so sorry.
It's beyond endosymbionts. Are there any other interesting organisms that have turned up in your trapping efforts and hemlock stands?
Oh yeah, we get all kinds of so one of the studies that Keely also did that I didn't talk about today is we have done amplicon sequencing where we've just taken those traps and looked at all the arthropod communities that we find and all the plant communities that we find. And because we're doing cytochrome oxidase is across most animals, right? We kind of get a lot of interesting animals that pop up. So mice apparently like to run across our traps a lot. So we'll get mouse DNA on there a lot. We get spider DNA, beetle DNA. We also, in areas where there are other invasive species, we've been able to pick up those. So we've been able to pick up spongy moth in areas that we know spongy moth happens to be in, even though the trap doesn't have spongy moth on the trap itself, like they landed on it and left. So I can't remember exactly how many families of arthropods we picked up on the traps, but I think on 100 traps, we picked up over like 75 different arthropod families of insects. So yeah, they're really cool. And one of the things that we were kind of looking at is, do we see like co-occurrence? If we see adelgids in one area, do we happen to see a co-occurrence of other species with that? And so we're picking up a few patterns like that. Like lady beetles, we typically find more lady beetle DNA in areas with HWA, which is interesting because lady beetles actually can feed on HWA eggs. And then spongy moth, this isn't the same way. And there's an interesting paper looking at spongy moths typically will infest areas with HWA because of how it impacts the hemlock itself. So yeah, so there's some interesting co-occurrence things that we're looking at with that data. So.
Great, thanks, Margaret. Sorry it was premature. But anyway, very interesting, wonderful talk, Charlyn. Thank you so much. Thank you everybody for attending.